and members of teams that are working again on the theme of rapid and urgency and participation. You guys can all see is just teaming throughout everybody who's working on this. It's my pleasure to continue this incredible conversation and follow up on presentations that you guys have hopefully already seen. And I'll remind you again to watch them a second time after hearing these incredible scientists talk to us. So right now it's my pleasure to bring up to the stage Doug Chapnick, who is the CEO and the founder of BioLoomics, who's another company and another individual, everybody who's in here. So Doug, thanks for taking the time.
Hi, Luke. Good to see you again.
Yeah, you too. I think there's a bit of an echo. Is that, are you on two devices by any chance?
No. All right, let me disconnect.
Well, it's better. It's 67. Think.
That might be the woes of where I live and the type of internet connection.
It might be a reminder for me to stop talking too. So maybe I can just jump into the questions that that came in. Thank you so much for that presentation that everybody saw. It was amazingly informative and had so many different layers on it. Looking at questions that are coming in, but I'm going to ask the first one was, if we were to explain how you're using your technology to find KAND treatment, this is a great question. If I were to explain it to my mom in 1 minute, what would I say?
Yeah, so I think about that a lot. We're a young company, and I'm a first-time CEO, so it's a big problem simplifying what we do. But so we have a platform tech that it has a mission to find therapeutics, right? And so one way to find therapeutics is to build a way to measure whether a drug is a good thing for a disease or useless for a particular disease. So what we do is we try to program cells to be fluorescent so that we can use them like bomb-sniffing dogs. So just like a bomb-sniffing dog at the airport, we still use that tool, and it goes out and the dog smells bombs out. But we don't understand how those dogs do that, right? We don't know what they smell, what their experience is, but we use them like a tool. And in the exact same way, we want to use fluorescent cells as a tool, so we can shrink these experiments way down and test millions to billions of drugs. And somewhere out there in the world is the drug, the idea is that somewhere is the drug for what we want to use in clinical trials for KAND. And the hurdle that we got to get over is the ability to know when we found it. And so once we have that, we just need to feed drugs in, right? And just do those experiments, find the winner, and then try it out in animals. And if it can get there, we put it into humans. And so our mission is to build that first tool that's rock solid, that when we see a cure in our cells, in our petri dishes, we know we're onto something. And hopefully there's more than one. Hopefully there's 20 or 30 or 100. And then we want to hand this off, right? So we build the assay, we find the drugs, hopefully other people, other pharma companies participate in that and feed the drugs into this whole pipeline. And then we hand them off to what you guys are sort of are building up, right? Another test. If it can get through all these hurdles, we know we're in really good shape. And so everybody sort of building their own test is a good thing, that we're not competing. We're hoping that the real drug will pass all the tests, and a person won't get a drug that doesn't work.
Yeah, that makes great sense, and it leads me to a question that we have all the time. We get it, and I wonder if you might just be able to educate the group about. You mentioned the word assay? Can you explain just to those tuning in what an assay is?
Yeah, it's a test. So it's some sort of thing you can measure and get a conclusion about.
Right.
And so if you have a drug, from like a layperson's perspective, a drug's just in a tube, it looks like clear liquid, right? But you have to, in order to understand what's happening with that drug, you need to put it into some system and see some change. And so we're building small miniature versions of these tests just so that we can sort through drugs, throw away the ones we think aren't good candidates to go to the next phase of development, and narrow this giant search space. So I want everybody to appreciate how big the problem is. There are so many possible ways to build a molecule that if you just scratch the surface and had one copy of each one of these, they would weigh way more than the Earth itself. So the possible chemicals that you could build have potentially never been sampled in the entire universe, right? So our mission is to somehow first find ones that are here and or create them and find this rare variant of how to make a molecule that somehow interacts with our cells and our tissues and our bodies to cure a disease, right? So that's true for all disease research. And for this one, it's no different. We really just need to find that rare thing. And in order to find it, you need to measure something.
That's great. That's a great description. Thank you. It leads me to another word people might have questions about, and I love this. And if you could clarify biosensor.
Yeah.
What's a biosensor? What is the KIF1A biosensor that you've created that you're sensing?
Yeah. So theoretically, we did create the sensor already. I should say that. We don't know that yet. But so what a biosensor is it's a fluorescent protein. It's a protein. So your cells in your whole body, there's only 20,000 proteins, and so it's a certain way to build a molecule. Each one of these proteins, they're chains of amino acids, and if that's complicated, you could really just think of them like Mr. Potato Head. They have different eyes, different ears, different hair, different shoes. And so what we do is mimic what nature does to build these proteins by taking pieces from nature and stitching them together with other pieces, and then building all different versions of proteins that we can then put into cells. And every single one of them is fluorescent. And our hope is randomly one of them senses something in a cell and that we can see that with a microscope. So our strategy to build these biosensors is to take millions of cells where each cell has a different version. All the cells are fluorescent. We just turn on KIF1A, so the mutant version gets expressed in every single cell, and then our microscope shows us every single cell's fluorescence. And one of those cells lights up. And then we go in and we vacuum that one up. And we take it and we find out what the sequence is. So we build millions of versions of these proteins, these biosensors, to throw away almost every single one of them. And one or two in every experiment does something. And so we expect we'll very quickly find candidates. And what we're looking for is really specific candidates that we can move forward through this pipeline.
No, this is really great. And there's two questions that lead kind of really well into that. And I could go, this is a conversation we could have forever. So I'm glad we're talking about it. But the first one is about failure. And Dominique speaks about this and spoke about this earlier. And we love the concept of failing 1,000,000 times to succeed a handful of times, right? And that was really just a point that resonated for lots of people I know. And can you expand just a little bit about why failure is so important for your project to be successful?
So it's just like there's a countless number of ways to build a molecule. There are countless number of solutions to build a biosensor, right? And so there's a rare biosensor design that's going to measure in cells what KIF1A does. And if we try to imagine which one it would be and guess, we'll probably guess wrong every time in our lifetime, right? So if we don't layer on random guesses and just try and do a volume game where we test millions in batches, and we know when we see one, but we're not going to spend any time trying to design one. We're just going to try to design as many Mr. Potato Head versions of this protein as we can and test as many as we can, and then image as many as we can to find that rare variant. So what we're doing is we're bypassing trying to understand how things work at a certain stage of the process. Not all of them. We understand how to put the proteins together. We understand how to recognize when we found one. But the middle part, we're ignoring that. We're saying that takes decades to 100 years to figure out. But we know we can randomly build these things. and we know we can find the ones that work. So let's just try and put all that together and forget about our sort of human need to understand everything and see what comes out of the pipeline. And drug development, it's always been like that. It's always been aiming for a lot of things, throwing them at the wall and seeing what sticks.
Yeah.
And research that goes into the mechanism is about throwing the right things at the wall. And so we're sort of balancing the two stories there.
It's such a great point. It's a point that, Dominique mentioned earlier, and we heard from Wendy talking about, failing fast and how that is, right? I'm always thinking about risk, fail, risk again, and do it fast, so we're not wasting any time. So that point you just made is so appreciated by every single person. It's so close to our heart, is that fail fast and risk again. So we really appreciate that concept. Now, I know I'm going to ask one more question, and it's about the translation part of things, right? So, 6 months to a year to finding a target is a great thing. What happens once you do find that target? What's the next step? How can we help? Who do you partner with? Who do you pull in? And what translates from discovery to product development?
Yeah, so we have lots of parallel directions going on right now. We just started, but the paths are clear. There's the path that some pharma company has the drug in their freezer. And that path requires us to somehow get to test those drugs. And to get those people to believe that it's worth handing over their drugs, and that we can work there's also the whole company part and proprietary part. So we have to get over those hurdles. Okay? So that path has some hurdles. Some of those might be FDA approved. And so they shorten the back end. That path's a good path, because we might not have to do certain preclinical trial tests on the drug if it's already FDA approved, right? So that's why we want to go down that path. It's more to see if we have some tool that we know a lot about that we could repurpose. And then the target path is a kind of complicated way we're applying our technology, but we're basically making a different version of Mr. Potato Head in the cells, this protein that we can scan through millions to billions of designs and find a design that somehow appears in our assay to reverse the effects, to basically cure KAND in those cells, and then pull it physically out of the cells and ask, what is it bound to?
Got it.
And when we do that, we get the name of a target. And if it's a target we've heard of before and we have drugs for, we're back in that territory where we could say, oh my God, we have a drug. I don't know why that drug would work here, but it looks like it's going to. And then we test it in all the different assays that you guys are developing as well, and then other assays. And so that's another path that we're taking. And that's where we try to repurpose the drug again. Okay, but then there's a third story. The thing that we pull out and the target that comes with it, the reason we did that was we thought it reversed the effects in cells. So our Mr. Potato Head could also be a gene therapy.
Right.
The thing you add to the cells, we found it because it cured the cells. So the question is it a viable path? And so we're a preclinical company. I want to point that out up front. Our mission is to stay preclinical, to do what we do really well, and it's our partners that will take them into the clinic. So at some point, when we have something valuable, that's when we go and we shop around, we find the right person that we are confident can do a fast track job of taking what we think is working, confirming it for themselves, and then designing this clinical trial. So we're actually if we're successful, we kind of pass the baton.
Yes. Thanks, Doug. That's a great point. I think it is the paradigm shift that's happening in biotech right now. So it's such a critical point. And there are two scientific questions I really want to get to from two really incredible people here. One from Michael Silverman and one from AJ. We'll start with AJ's, which is how will a bioassay based on kinase activity help solve a problem related to anterograde transport?
That's a great question.
Yeah.
It requires a little bit of stepping back from the current paradigm of how mechanistic research has both limited and accelerated drug development. So a kinase that we're using assays that are like beacons in a network. So if they measure a downstream effect, that is irrefutable in the cells. We picked it because it responded to a particular turning on KIF1A mutation, right? So we know that that is true, that biosensor is somehow coupled to the network change, right? That why is that happening is sort of inconsequential to how we're going to use it. So we're using it only as a beacon to go out into chemical space and narrow down the search space to a few testable compounds that can go into other assays, right? And so ultimately those other assays will be the validation.
Right. Got it.
And so the short answer is almost every molecular pathway for every disease has a kinase in it. Even though the question was more about this piece of a pathway, how a sort of cellular machinery is moving down a microtubule. There's a molecular pathway sewn into the beginning and the end of that, and there's kinases there, and there's feedback loops that bring it back around. So you could basically measure a butterfly flapping its wings and see a storm happening in the cell, or vice versa. And so it's not about saying it must work a particular way. It's about recognizing when we find that beacon and then utilizing it to move fast. It's a very different way of thinking.
And similar question to that Michael's is a good segue, is about cellular phenotype and what you're assaying, right?
Yeah.
So, he mentioned with the biosensor, ATP productivity, production or motility, things like that. So what are you, if you don't mind saying?
So what ends up happening in these experiments is usually we find many pits, and they're all built to measure a kinase in a particular pathway, and so it's sort of like, again, it could be working in mysterious ways. It sort of doesn't matter how it works. What matters is that we pick the assay that has the most power to narrow down the search space of drugs that will be successful in the next tests. So the way we're going to do that is we're going to compare which assays are harvested from each mutation of KIF1A against the wild type. And when you see ones are common across many mutations, then we know we have power, a sort of replication of the biosensor development that's not reproducing for the wild type. So what it says is that the biosensor is measuring something that happens that's very specific to the mutation. It's not specific to the expression of KIF1A. It's specific to what the mutations do to the cell.
Right.
And so it's out there. I'm not predicting how the biosensor is going to work. That sort of falls out later in our pipeline. So we've done this 5 times so far, and they're all in various phases of a little bit past proof of concept, and some of them are striking deals right now. So it's sort of a fast-paced, very just emerged thing that we're doing. The mechanistic part has yet to come.
It's really exciting. Doug, I can't thank you enough. I encourage everybody to go back and watch Doug's presentation 5 or 10 times, because it's really fascinating and really just exciting and inspiring. And again, has that theme of urgency and teaming with, just incredible participation from so many people. And Doug, your work means the world to us. I can't thank you enough. If there's one message you might want to say to all of the families tuning in, is there something that you might share?
Yeah, I mean, I am personally blown away by the conference and the organization. And it's the first time I've ever been exposed to something like this. So I think all of you should be fairly proud of what you've created. And I truly believe you're on the mission and that will be successful, and that it's by you all coming together that will coalesce the resources to solve this problem. And so I mean, I hope the pride is being felt, that you're sort of the nucleating point around which this will come to fruition.
Thanks, Doug. Thanks for for partnering with us and to your whole team at BioLoomics, and thanks for climbing this mountain with us. We're going to get there fast together. So I really appreciate your time and your presentation, and I'm sure we'll be sending back pages and pages of questions that I'll ask you.
Oh, yeah, please do.
So really appreciate all you do, Doug, and thanks for being here and joining us at this conference. It means the world to us all. I can speak on behalf of the entire community. I really appreciate that. Thanks, Doug from BioLoomics.
Thanks, everyone.